Hello, everyone, and welcome to NOAA's Omics Seminar Series. My name is Tracy Gill, and along with Catherine Egan, we are the webinar co-hosts for this series. Before we get started, here are a few logistics, also listed in the chat box. For this webinar, the presenter is willing to address your questions, clarifying questions during the webinar and mostly at the end of the presentation. All attendees are muted, but feel free to type your questions and comments into the chat box at any time. If you want to go to full screen or a larger view, find the box at the upper right of the slide deck and hit that box. This toggles full screen view on and off. If you're interested in getting a copy of the recording, I will send them out a day or two after the talk to all who are registered. If you are not on the NOAA Weekly Science Seminar email list, but you'd like to be, please contact me at tracy.gill@noaa.gov, and I will add you to the list. And thank you to our captioner for making this webinar more accessible. And now I will pass the mic to my co-host, Catherine Egan, Omics Coordinator in OAR's Ocean Portfolio with the OAR Office of Ocean Exploration, and she is also the subcommittee co-lead for the NOAA Omics Working Group. Catherine will introduce the NOAA Omics webinar series and today's topics and presenters. Catherine, the floor is yours.
Thank you so much, Tracy, and thank you all for joining us today. As Tracy said, welcome all to the NOAA Omics Seminar Series. As Tracy stated, I am your co-host, Catherine Egan. The Omics Seminar Series is hosted by two NOAA groups, NOAA's Ocean Exploration and the National Ocean Service through the One NOAA Science Seminar Series. So omics is a suite of tools that are used to analyze DNA, RNA, proteins, and metabolites, and has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. We have a brand new email address, which I will drop in the chat in just a moment. So if you have any questions at all about our seminar series or about the omics portfolio across NOAA, you can email noaa.omics@noaa.gov. So with that, our speaker today is Dr. Ole Shelton, and his presentation is titled Environmental DNA Provides Quantitative and Efficient Estimates of Hake Abundance and Distribution in Support of Fisheries Management. So Ole Shelton has been a research ecologist at the Northwest Fisheries Science Center since 2012. He received a PhD from the University of Chicago in 2009 and was a postdoc at UC Santa Cruz. He is a quantitative ecologist who works on a range of marine systems, from coastal eelgrass and kelp forest communities to Pacific salmon and groundfish spatial distributions and dynamics. He both develops and applies new statistical approaches to large data problems in fisheries and conservation. Since 2014, he has worked on both single and multi-species applications of environmental DNA technology in estuarine and marine habitats. So thank you so much, Ole, for presenting today. I will now pass it off to you.
Great. I think I'm unmuted. Thanks, everybody, for joining. It's morning in Seattle. It's 9 a.m. So still drinking my coffee. I hope everybody else is too. Today I'm going to be talking about a very large project involving a lot of people about a sort of a kind of a new application of a lot of omics stuff. And this is sort of like treating omics essentially, or environmental DNA in this case, as something more akin to traditional fishery sampling. So sort of using omics in a weird and unusual way that has sort of come about in the last 10 years. So move this over to the side. I like to start by sort of giving a big picture. At NOAA, we're interested in counting fish mostly and understanding the ocean ecosystem broadly. And sampling the ocean is hard, right? And we do it in a bunch of different ways. We use big boats in rough seas and drive them around. We dive in kelp forests and count things. We use nets, like down here in the lower left, to sample fish in the near shore, and we go into the deep sea using submersibles, right? And these are all different methods. They're all different ways of looking at the world and trying to figure out what's going on. They're all imperfect, and they all have interesting biases and attributes. But they're all sort of based around this basic question of, how many fish are there, or how many whatever species you're interested in are there, when were they there, and where are they, right? So it's just basic, it's fancy counting by and large, but it's fancy counting with a purpose. So the notion of environmental DNA is essentially, here's this beach shore, beach seine crew catching fish. There's 6 people. They can do 6 or 8 or 10 beach seines a day, and maybe environmental DNA provides an easier way than directly catching a lot of fish. So instead of having 6 people and lots of work, you can send my friend Jimmy here out into the ocean or out into the near shore here and collect water. So maybe we can survey creatures without catching them or seeing them, and maybe we can do it faster, more efficiently than we traditionally do. That's sort of the promise of environmental DNA. A very short primer on eDNA is that everything sheds stuff into the environment as it lives. So fishes are swimming around, they slime slides off them, the skin sloughes off, or the scales, excrement, whatever. They're dropping stuff in the water as they swim around and do their daily thing. And the other part of this is that the DNA and various cells associated with that sloughed off material degrades. And it degrades relatively quickly. Scale of hours to days, depending on your habitat and other variables. And so together, you put those two things together and you simply say that, well, samples of the DNA collected from water should tell you something about what is nearby. And clearly there's some complexity here. What does nearby mean? How much stuff gets shed? How fast do things degrade? So there's details, but the big picture is this should tell us about local or relatively local information about what is swimming around and how much of it is swimming around. So we like to talk about eDNA as a new kind of net, right? And so just for comparison purposes, this is a beach seine, and we more or less know a lot about how this works because people have been beach seining for, hundreds of years. We understand the characteristics of this net. We know how much area is being sampled about it. We know which mesh size, what's being caught or not caught. We know the depths that are sampled. And there's sort of an immediate way to translate what we see in this net to estimates of abundance or biomass or other things that are of management relevance. So if we're thinking about eDNA as a new kind of net, here's Jimmy again collecting water. What about this net, right? Do we need to worry about the temporal and spatial variability of DNA? Do we need to worry about it being transported or produced in different places or degrading at rates? And how do you take this from just a water sample which collects some DNA and translate it to abundance or biomass or other things in the units that we want to talk about? So a lot of this whole sort of field of work, in my opinion, is talking about characterizing the eDNA net and figuring out what we can and can't use this sort of new technology for. So this is part of, this work is all part of the larger NOAA Omics initiative. And for our point, this is sort of has 3 main parts. One is trying to figure out, can you use it to develop indices of abundance? How do you, and what sort of attributes does it have relative to traditional surveys? And then trying to apply it in new situations. So understanding abundance of fish communities for other species, both either in a single species way or in a multi-species way. I'm not going to talk about other people's work very much, but I just wanted to point out there are a lot of people trying to do this in a lot of different places in the world, and also lots of different habitats. So rivers, lakes, near shore, offshore, all these places. This is a larger and burgeoning field, and a lot of people are trying to figure out how to use this data in an interesting way and use it in and make it practical and efficient for management. So what I'm going to be talking about is what I call going big, which is, I think, to my knowledge, the largest eDNA sampling in the world at the moment. And this is looking at Pacific hake. And so for those of you who don't know what Pacific hake is, It is the largest ground fishery on the U.S. West Coast. We're talking hundreds of thousands of metric tons annually, and very valuable as well. So millions of dollars, certainly many millions of dollars annually in ex vessel value. And why are we working on hake? Well, it's a big species with a lot of data. It's important for commercial and ecological reasons, and it also allows you to contrast what's going on with eDNA with a whole bunch of other methods. And I think of hake as a test case for general applicability. If it seems like it's doing reasonable, good things for hake with a species we know a whole lot about, it can be ported to other species that we don't know very much about, and maybe eDNA can reveal all sorts of secrets or information about understudied or underappreciated species. So I mentioned we talked a lot about spatial, or know a lot about hake, because there's a giant survey for them. So this is generally a biannual survey, so 2017, 2015, 2013. And this is an acoustic trawl survey. So this is just results from the acoustic trawl to orient you. This is Southern California, it's Point Conception, San Francisco Bay, the Columbia River. So this is the West Coast. And in these plots, red means there were a lot of hake. They saw a lot of hake in that survey at that point. Black means they didn't see any hake. There's details, of course, in there, but the point is some years there's a lot of hake. Some years there's not quite so much hake. And the distribution of it shifts a little bit from time to time. It's generally sort of, there are places that seem to be consistently lots of hake, but things move around where they see the 3 years, right? So what we're interested in is, basic question is, well, does the DNA look like what we see from acoustics? That's one major question. A little more details on the survey is, this is a survey designed in 2017. If we zoom in, the acoustics are these black lines right here. And so they're going along latitudinal lines. And then at each of these X's, they sample a CTD. So they dump a CTD off the side. And in 2019, the Hake cruise was, very generous in providing help. And at night, we collected water. So every time they dumped a CTD into the ocean, we collected water that could be used and filtered that could be used for eDNA. We didn't we didn't get water from the entire survey extent. I'll show you the subset in a few minutes. But we have a lot of water and a lot of samples. This is the survey extent here. So we're from just north of San Francisco up to the Cape Flattery, which is sort of the Washington, Canada border up here in the north. And at each of these CTD stations, we're sampling 2.5 liters of seawater from multiple depths, so 3 meters, 50 meters, 100 meters, 150, 300, and 500. There's several, we're ending up with about 2,000 samples. These are taken at night. It's important. And of course, it's CTD, so they're taking all sorts of other associated oceanographic data as well. And there's some controls and other things that I can get into later. Of course, in parallel with this, during the day, the acoustic trawl folks are working really hard, and they're sampling what they see with the acoustics. So that involves driving the boat along these transect lines, looking at the echograms, taking midwater trawls to get age and length information for hake, and these producing estimates overall, and this is sort of summarizing an enormous amount of work, is they're producing estimates of depth integrated biomass, or depth integrated biomass density, so metric tons per kilometer squared. And we're talking about, this is an estimate based on age 2+ fish. I see there's a question here about what type of filter. I forget off the top of my head, but these are large filters. So these are mostly, we're trying not to catch plankton. So this, I think it's 1 micron, if I'm correct. Some of my collaborators are probably online as well. Could correct that. And I will show you a reference to the more of the details on that in 1 second. So that's that. It is important also that we we are talking about age 2+ fish, and that we can come back to that into the discussion at the end. So we're not talking about very small fish, juvenile fish, eggs, things like that. All right, so what I'm going to do is I am not, as perhaps is evident from my last answer to the typed question, I don't spend a whole lot of time in the lab. My job is mainly taking the excellent laboratory work that people do who are my collaborators and sort of using it downstream. So this is another point of this general topic, is that you can't do it alone, and no one person has all the expertise for this. So I'm more on the quantitative, what do you do with the data side? And we have really good people who we can give you all the details you want about the lab side as well. Anyway, going back to the talk. So the general approach here is, let's try and make two surveys. We have an acoustic survey. I'm going to treat the eDNA very similarly to how I treat the acoustic trawl data, and treat them as independent surveys and figure out, do they give you sort of interesting or similar patterns in what you see. I want to look at what goes on at about the population at multiple scales, so the local scale, as well as sort of a broader population scale. And for me, the big thing here is going beyond sample-to-sample comparisons among methods, right? There's very good reasons that when you take a bottle of water next to a net that caught fish, those shouldn't be the same. And I think doing that comparison, nobody really cares what you catch in one individual sample. What you care about is what your inference is at the population level. And so one of my big sort of hobby horses, if you talk to me outside of the seminar, is stop doing sample-to-sample comparisons, because mostly they're not very interesting, and they don't tell you what you think they're telling you. So we're going to try and not do that as much as possible in this talk. I only have a schematic of the eDNA workflow. Anna, who just answered some questions in the chat, has a paper that is just accepted, I believe, down here in the corner. We are happy to share that with anybody who's interested. Schematically, this is what we're doing. We have our CTG here. We collect water in 2.5 jugs. You cell capture it by using vacuum filtration. You preserve it. And then take it, that's all done at sea, almost immediately after you sample it. And then we take it back to the lab in Seattle, extract it using the really unpleasant phenylchloroform extraction because it gives lots of DNA, and then use qPCR primers to amplify Hake. So we have Hake. We also have done a couple other species, which I'll mention at the end. This talk is all just going to be about Hake and qPCR. And so we can just jump into some results. So this is more or less raw, close to raw data for what we have for Hake DNA by depth. So each panel is a depth. So here's the surface, 50 meters, 100 meters, 150, 300, 500, and then the spatial series, right? So, you have a lot of red pretty much everywhere. So the point here is everywhere you see red means there was detectable amounts of Hake DNA in that sample. So one conclusion here is Hake's everywhere if you look at the eDNA, right? There's variations. There are some places that clearly have a lot more DNA. Size of the circle means more DNA. There are some places that clearly have not very much DNA. But big picture, there's an awful lot of Hake in an awful lot of spots. Another way you can look at it is you can sort of take slices of those transects. So this is at a particular transect. So this is latitude 38, which is California, Northern California. Here is the shelf, the sort of brownish color. And then each of those little dots is where we did a CTD, where you collected Hake eDNA concentration. So again, bigger circle means more DNA. And this blue is the associated estimated Hake biomass on that transect. So you can sort of squint at this one. You can squint at a different one from a little farther north. And there's clearly patterning. One, there's clearly patterning in where you see DNA. Generally speaking, it tends to be over this continental shelf break. Generally speaking, it seems to be between 100 and 300 meters, roughly. That's just sort of a raw data, sort of squinting at the information. We can do the same thing for the acoustic trawl information. So this is derived directly from the Hake crew. And so we can say, well, where did you see at least one hake? So green means you saw some hake. Purple X's mean you didn't. And then also how much, right? So big circles mean you saw more hake on the acoustics. Small circles mean you saw just a little bit, and then empty spots mean you saw none, right? So there's clear spatial patterning, just like I showed you from other years. So just squinting at the raw data, we already have some sort of clear differences between the eDNA and the acoustics, right? So the eDNA says hake are almost everywhere, right? More than 90% of samples have some sort of hake in them. Many are very low concentrations, but you detect them pretty much everywhere. Acoustic data says hake are very, very patchy. Only about 30% of the segments are non-zero, and sometimes they are very non-zero. So there's very high densities in some of these places. I will come back to this at the end, but there is that 90% should be sort of accepted a little skeptically, because there is small amounts of contamination that show up, and it doesn't really matter for most of the things we're doing, but for percent of samples that have fake DNA, it probably bumps that number up a little bit. So, right, so graphically, if you wanted just to sort of think about the attributes of DNA versus the attributes of this acoustic trawl survey, is like the acoustics are really peaky, right? So you have giant piles of hake in particular spots that you see, and then long patches of zero, so they're up and down. eDNA is sort of more like these smooth rolling, verdant hills in this picture, right? So everything's sort of smoothed out, everything's kind of not too peaky, and these, from a sort of statistical sampling perspective, these two patterns have very different downstream consequences for what you're going to do with the data. So how easily you can estimate total population size are dependent on what this distribution looks like. So that's interesting, just to begin with. All right, so that's the raw data. I think it's from raw data to something useful. So what I'm going to do for the rest of the talk is sort of build some spatial models for these methods, make projections, do some aggregation, and then do some comparisons at the very end. Clearly, there's a bunch of math here I'm not going to tell you about, but I'm going to try and nod to what I'm doing. I'm doing this in a full Bayesian framework. I'm making a state space model for DNA and another one for acoustic trawl that are independent. For the DNA, I have a process model that involves some true but unobserved state. I'm allowing that state to be smooth in space and as a smooth function of some covariates, namely bottom depth. And then I have a pretty complicated observation model, which accounts for a range of water processing issues. So sometimes you had some inhibition, you have to account for that. Sometimes there's small amounts of variation in how much water was filtered, you have to account for that. We have replicate qPCRs for each sample. We have replicate water samples from each CTT. We have standards for the qPCR. So there's a bunch of stuff in here. Most of anything that's different from a lot of traditional fisheries models. And this observation model turns out to be interesting and not what most fisheries folks would think of in a lot of cases. The acoustic trawls is, I'm not afraid, I see a picture from Kathleen. I don't know what DVM is, but maybe you can type that out. I can answer. For the acoustic trawl, we have a hurdle model. So this is sort of, we have a model for occurrence, we have a model for positive. And for each component, we do the same thing that we did for the state space, or for the eDNA model, where we have a smooth bottom depth, a spatial smooth, and they are independent, and we multiply them together to get a prediction across the whole space. Diel vertical migration. Diel vertical migration will move the hake around a little bit. It'll bump them up. It'll bump them down. But they're still in the box that I am interested in, which is the whole West Coast. So I would think Diel vertical migration would smooth out the signal vertically, just like I was talking about it for being smoothed out sort of spatially. Again, there's a lot of technical things here. Models fit pretty well. They converge. They pass basic tests. And the model results are relatively robust to various changes in model construction that I've sort of poked at for the last, few months. And that includes changing what the main terms are, changing how smooth things can be, and then putting in alternate observation distributions. People who want to talk about the technical details, drop me a line. Happy to chat with you all day about them if you want. So we can jump into the results. So we went from raw data and we fit a model, and now we have sort of these projections. So, and this is the form of some of the results are going to happen. So we have sort of a field at 3 meters, which is the shallowest area. We have a field at 50, 100, 150, 300, 500, right? Dark colors mean not very much DNA. Light colors mean lots of DNA, right? So this is a square-rooted scale, so there's a big change from very low to very high, so 3 orders of magnitude. And what you see is that there are pretty clearly depth-specific patterns in DNA. So mostly, the highest stuff is sort of in this 150-meter stuff. So there's a big chunk in Southern Oregon. There's a big chunk along the shelf break down here in California. Similarly, there's high levels here at this 300-meter area. There's also a lot in Cape Mendocino. You get down to 500 meters, everything's sort of dark and sort of boring. You get some interesting patterns up at the surface. And that's interesting because we don't think of a lot of hake being at the surface, except for potentially bringing down Diel migration at night. So, the acoustics would never see them at the surface, even if they are there at night. So there's some interesting questions there. We also see really interesting patterns in uncertainty. So we can talk about this in terms of the coefficient of variation. So again, there's 6 panels here for each depth, and these two shallow areas have a lot of uncertainty in CV. So it's really, really high levels. It could be all sorts of different values at the surface and at 50 meters, and it becomes more predictable as you go down. Another way to plot this is just sort of averaging across all of the space. So you have very high CVs at 3 meters, at 50 meters, and it sort of falls to about, a reasonable level, about 0.3 for 100 meters and deeper. And so this is driven mostly by between bottle differences. So 2 sides of the CTD have substantially different values for the DNA concentrations, and also small scale site variation. So one CTD site is very different from a site right next to it. And that's interesting. I didn't expect this. I don't really understand what's going on, but it's certainly a pattern that is undeniable in the data. So super interesting, and has ramifications for how you might think about what you sample in the shallow or relatively shallow waters of the ocean. You can also plot these things by depth, so we can talk about where do we see the largest hake concentrations, and these are sort of at moderate depths, sampling depths, which is sort of this y-axis here. So this is reversed axis, so this is the surface up here. This is very deep, and we always see peak DNA sort of in this 100 to 300 meter range, and you see it over the shelf break. So this is where the different colors come in. So this is 150 meters when the bottom depth is about 150 meters. This is the sort of darker blue area is just a little bit deeper on the shelf as the shelf goes down. So this is the peak spot. And so this is along the continental shelf break in moderate water depths, which is exactly where we expect to see hake based on published estimates and talking to the Hake cruise folks. Yeah, so that's where we're at. So this feels good from sort of a intuition standpoint that this is where you expect to see hake. So another relative to the diurnal migration. So maybe this is where everybody thinks hake live. It seems the DNA shows pretty clearly that that's where the hake are hanging out. And then maybe some of these things up in the surface are related to, you know, their migration at night, where they don't spend as much time there as they do in the main part of the, or their sort of core area at 150 meters or so. I see another question. Do you make results available for other species? Yes, I will. That will be, wait till the end. I'll talk a bunch about that. But yes, there's lots of interesting things that we can do with some of these samples. So that's great. So that's sort of a description of what the DNA and the model fits look like. The other thing you can do is you can start aggregating things and synthesizing them. So this is a integrating DNA from the water column to generate an index. So we're going to use the full posterior from all these different slices of depth to make a full index from between 50 meters and 500 meters. So we're excluding those surface samples because the acoustics mostly don't see those very shallow waters. And this is the mean index, but we also have uncertainty about this index as well. So, and you can see this is sort of a pattern that looks ballpark like those Acoustics series that I showed you earlier, right? So there's a lot of fish along the edge. That's the continental shelf break. There are little hot spots. So this is in sort of central Oregon, like I talked about before. There's sort of a lot of fish predicted to be here near Cape Mendocino and south. So there's clear spatial patterning. And let's see, we can do something similar for the acoustic trawl survey. So this is spatial model results. I'm not going to go into as much detail about these, but we have our probability of occurrence model. We have a conditional density model. They both fit okay. This conditional model is hard because it's very noisy. So there's a lot of uncertainty about it. And we can come up with a projection that are on the same grid in similar units. So this DNA index is not in metric tons. The biomass estimates are in metric tons. And if you look at this, you see a little bit and this will be a little bit there's a little bit of difference here, but you do see strong areas of correlation. So for example, the 6 area, there's a big, predicted to be a big pile of fish in this box. There's a lot of DNA there. A fair amount of DNA in this box. There's predicted to be a lot of fish there. Right, so there's sort of pairwise correlations. I see a little bit of questions here. One from Graham, could DNA at surface be coming from fishing vessels? Yes, they could be coming from fishing vessels, but I don't think that's the main component. There are no fishing, or very few fishing vessels in California for hake. There's some, and it's not necessarily the case that most of these, the hake survey doesn't necessarily get near or close to any of the fishing fleet as it is. So I suspect it might, that you can imagine that affecting a little bit. But generally speaking, that shouldn't be a big component of what we see in the DNA. Sarah, number 2, do you account for DNA molecules drifting due to vertical upwelling and horizontal currents? I don't. I think because I'm not really concerned where they came from. I know that they came from within the spatial domain that I'm interested in. And I know that based on DNA degradation rates that they did not The Hake did not come from outside my box, so there's going to be a little bit of imprecision. So I cannot track which DNA molecule corresponds to which fish, and I am okay with that because that's mostly not. It's sort of a smaller scale than I feel like I need to know about the details for where the DNA came from. Diana, number 2. To add to Sarah 2, is there evidence that you're picking up hake from predator scat that could be adding to the noise at some shallow depths? I suppose so. I hadn't thought about that. I don't think we have any evidence one way or the other along that. Amy number 2, along with your salinity question, have you looked at, I must have missed a salinity question. I don't believe the salinity really, this is all open ocean. The salinity is pretty consistent until you get down below a picnic line. There's not a huge amount of variation. So have I looked at UV degradation of DNA through the water column? I have not. That is partially why I exclude surface samples, because I am less worried about DNA degradation 50 meters down and deeper. And finally, we have a question from Alyssa 2. Have I noticed any differences in location between trawl surveys and DNA collection? For example, could fast-moving water or upwelling cause slight shifts in where you see hake versus where DNA is found? Yep. The notion that DNA moves around relative to where the fish are is totally reasonable and interesting, but I'm not that interested in sample-to-sample comparisons, mostly because I don't think that's the scale that we should be worried about things. And so I'm interested in at the sort of coast-wide scale or at the 5-kilometer scale that we're talking about here. And on those scales, those sort of small differences in where things move around are sort of get averaged out. So do I believe that the DNA distribution in the water column is different than the actual hake distribution? Yes. Do I think it changes what I'm interested in, which is population estimation? Not really. So anyway, that's my feeling on that. And all right, here we go. So I can move back to the talk, and you can say, do they provide the same answer? And we can do pairwise correlation. So this is at this 5K cell, or we could plot acoustic biomass against DNA index. And, you sort of feel good about it, like it's positive, but there's a lot of scatter. So there's places where you see a lot of hake in the acoustic trawls, and not very much DNA, or sort of a middling level of DNA. There's also places where you see a lot of DNA, sort of middling acoustic biomass. So it's not a perfect relationship. You do have these interesting sort of different distributions amongst the different, or within the different methods. So for the acoustic trawls, you have this very skewed distribution where you have a lot of things that are 0 or very close to 0, and this long tail where occasionally you see huge numbers of fish. For the DNA, you don't really see that. You sort of have a bunch of places that are low, but you have a pretty clear peak, and then sort of this sort of longish tail. They both have a long tail. This one, the Hake, the DNA one, is much better behaved in terms of estimation than the sort of acoustic trawl plot. So that's at the sort of 5K across all of the different whole domain of the survey. You can also sort of bin things up into these 1-degree bins. So these are you can probably see it down here in the corner, is that there's these sort of horizontal lines, dashed lines, and they're numbered. So 1 is in the south, 11 is in the north. So here's number 11, that's the most northerly plot. And when you do this plot, when you're aggregating up to larger and larger scales, things become very, very similar in a sort of a shocking way. So your correlation is like almost 0.9, 0.87. And that's sort of amazing, right? So essentially it says, if you don't worry about these small scale things and you move up to larger and larger scales, you're getting the same information, right? So the information from your acoustic biomass lays perfectly on top of your DNA or vice versa, as long as you're not worrying about these sort of very small scale deviations. In aggregate, which is what most stock assessments, most fisheries sort of things are based on, which is the population scale, this is what's interesting, right? So suddenly what looks like something that doesn't really match onto each other becomes very reasonable at larger scales. And this is sort of as a sidebar, when you divide it up into different boxes, you get more or less the same area. So if you use slightly larger boxes, you get close to the same answer. You use slightly different lines dividing the boxes, you get the same answer. Anyway, so this is a pretty general result, and also very similar to some stuff that we've done historically with salmon in estuaries. So I just said most of this, but they're almost identical at large scales, which is, you know, almost, it seems like black magic in a lot of ways to me. I don't really quite get it. I'd also point out that this happens even though your CTD stations are sampled more rarely than your acoustics. You know, we're talking about 186 point samples for the CTDs instead of 4,000 kilometers of acoustic trawls. The uncertainties are about the same despite these two sample size differences. CVs are about 0.1, which seems a little low to me, and I need to work on that a little bit more. So big picture, eDNA seems like it's providing reasonable information, and sort of the downstream effects of this is eDNA might provide a second survey without any more days at sea, right? So you're getting a second survey at night from the acoustic survey that you're doing during the day, which is, you know, fun. You can do other things. So the similarities in these two methods also give you similar patterns in species distribution. So here is the center of gravity, which is sort of a standard spatial distribution estimate for acoustics and eDNA, and they both pick out the exact same spot with, you know, slightly different error bars. You know, if you look at the cumulative distribution for these two things, you know, one is for the DNA, one is for the acoustics, and these sort of ribbons overlap each other the whole way up. So if you're thinking about what is the distribution of the fish in this spatial box, they're telling you the same answer at these sort of larger scales. And I already told you about the depth, which maps pretty closely onto existing published estimates. So those things are all very encouraging. So I'm just going to wrap up and talk a little bit about some of the things that are coming next. So I told you about this big eDNA sampling, lots of kilometers, lots of depth. We did it already again in 2021. Our intrepid tech Abby is out sampling as we speak, and we're planning on doing it again in 2023, so we'll have a time series. I do think this demonstrates potential applications to eDNA in lots of ocean scale, large scale ocean management problems. You know, the eDNA is consistent with Hake biology and distribution. It provides similar indices of abundance. It's a second index of abundance, maybe, for the same number of sea days, which is amazing. Because sea days are very expensive for all you people who work on boats. And the spatial scale of what we're talking about is comparable to lots of big fisheries worldwide. So large fisheries are, you know, on the 100,000 kilometer squared area or more. And this is sort of a first test case for that, you know, large scale application. Caveats. eDNA is not a panacea for all your ills, right? So it doesn't give you information on age, length, fecundity, condition, diet, disease, other things that I've probably forgotten. And that's important because those are all big parts of how we manage and how we think about fisheries. Surveys are largely valuable as time series to reveal relative abundance trends, at least in the fisheries world. And you need to have relative abundance to do this kind of thing. So it'll be years before eDNA provides multiple years to compare against through time. And so together, these things suggest that eDNA surveys are a supplement to existing surveys, not a replacement. At least at present, and maybe the future will develop more technologies. Like I said before, Hake is a proof of concept species. This goes back to the earlier question. So understudy species are likely more valuable, right, for uses for this. So Eulicon in our case is lamprey, threatened rockfish, marine mammals, they were brought up earlier. We're using these same samples for a number of metabarcoding projects, understanding fish communities with depths, relationship to oceanographic variables, and other things. And like the person noted before, these archive samples can be re-examined for other species in the future. And just to point out that we are doing this, I put in one slide for Ulicon, which is either threatened or proposed as threatened along the U.S. West Coast. And these are the same sort of panels I showed you before for Hake, but they don't look anything like Hake, right? So this is the surface again, 0 meters, 50 meters, 100 meters, 150 meters. X's mean there is no detectable eDNA. And we have this really clear pattern of really patchy eDNA in right near the Columbia River mouth. Right, so lots of DNA there. At the shallow, there's sort of small little pockets of Ulicon at these various other depths. But it doesn't look anything like Hake, right? And there's like lots of more variability than Hake is. And so there's a good question about, how general are the results for the Hake? How do we think about what is an expected amount of smoothing or not smooth distributions for Ulicon relative to the other species? What would a marine mammal look like? All sorts of questions are interesting. And moving on. And also there's a lot more technical stuff to work on, right? So the spatial models for acoustics and eDNA are fine, but they could be better. We need some more sophisticated depth comparisons. There's real interesting things going on with the observation models for eDNA. That eDNA is just different than most other applications. In the spirit of some of the questions, and I thought about this, do you really need to worry about the explicit dispersal or degradation processes? I'm not convinced you do, but certainly an open question and certainly a big question in the literature. And I've also sort of dodged this question about what about juveniles, other young stages that you should be able to see in DNA. My suspicion is that most of the stuff that's going on up at the surface is small juveniles, eggs, and whatnot that are not seen by the acoustic trawl survey. But that's just total speculation. So with that, I need to say thanks to everybody. And there's a lot of people, like I mentioned at the beginning, lots and lots of collaborators doing lots of interesting things. And I'll leave you with a picture. And unfortunately, this is not my email address. We made a mistake here, but it should be ole.shelton@NOAA if you want to track me down. But you can do that. I've seen a lot of things come up in the chat, and I've been ignoring them. And let's see if I can find a place where I left off. So I see a picture from, or sorry, picture. I see a question. I think I left off at Alyssa at number 2. And I think Ian, seems like vertical migrating hake, fishing vessels, predators, and currents are moving slow enough that you won't bias your overall picture where hake are if you aren't too focused at the fine spale. Yep, I 100% agree. That's the hope. As long as you don't, I think the key thing is how fast does your DNA degrade. So if you don't care about the scale of hours to days, which I don't because this is an biannual survey, I think what you're seeing is a reasonable description of the population. I see Amy too had a question, and Anna and Krista successfully answered her. Andrew, number 8. Any plans or advantage to creating an integrated eDNA acoustics model? Perhaps there is. That is part of the, so there's an open question, and this is something to talk more detail with the stock assessment folks about. And right now, I think of it as you make 2 indices and you feed them both to the stock assessment, and that's what stock synthesis or other stock assessment programs do, is they sort of deal with how do they conflict or not. For other kinds of questions or other applications, you can certainly imagine doing a full integrated data thing. When you do, or full integrated model. When you do that integrated model, however, you need to be careful about you start needing to worry more about these degradation questions, right? Like if I see a sample here for acoustics and I see a sample next to it for eDNA, how do I worry about if they talk to each other? And so that's where those questions about process start to matter a lot more. So short version is what you're proposing is super interesting and super hard, and we haven't figured out how to do it yet. From Wes, how is this going to be an integrated assessment? At present, it is not. This is full research, and we are talking to the assessment folks and trying to figure out what it would be reasonable for. Like I said, it doesn't really become valuable for a few years, and so we have time to work on it. Do I think it'll provide different data? Perhaps. I'm not really quite sure about that. How does it compare with cost-wise to acoustics? That is a good question. Most of the cost for the survey is in chip time, right? So you're adding something here that doesn't cost that much. It certainly costs people time to be sampling the water and people time to be processing. This is something that Krista and I should sit down and pencil out and do a full accounting to be more upfront about what it costs or what it doesn't. If less time in the field, it's more time in the lab. All right, let's moving down to lots of questions here. Okay. Should I just keep talking, Catherine, or should I start typing? What would be the best way to go here?
You can go ahead and answer the questions, Ole. Maybe it'll help if you could read the question out loud.
Sorry, I'm trying to. I go into my brain when I'm reading.
No problem. Yeah, a lot of questions came in when you were talking.
I see that Wes has answered Andrew 8's question, so I'm going to skip that one. Tracy 3, what is the cost to run an eDNA sample? That is another great question. I sort of dodged it before because I don't have a hard number for you. So it sort of from the, for our purposes, the most expensive part is having a person on the boat collecting, and then you have one person in the lab processing things. So per sample, there are scales of, there's massive variation in efficiency with more samples. So if you can do things, 15, 100, 200 samples at a time, you can get your cost per sample pretty low. If you're only doing 1 or 2 samples total, that gets more expensive. So anyway, interesting. I don't have a number for you. I apologize for that. Kurt, number 2. This is great. Is the survey sampling at the same frequency this year as was done in 2018? The interesting to see power analysis of how many samples and at what longitude and depth you are needed for a target CV. I suspect this varies from year to year, and the sampling is constrained by available time during nighttime ops. I think you're right. We actually turned out to get more samples this year than we did in 2019 because the Canadian boat that was supposed to take part of the survey in Canadian waters caught on fire, and so it turns out the NOAA ship went from Point Conception north up into British Columbian waters. So we have a bigger spatial scale. We have ballpark the same pattern of CTDs, ballpark same patterns of depth. So I'm super curious to see how similar or different it is from the previous samples. I'm not sure how it's going to turn out, but I'm excited to look at it. Yes.
Hey, Ole. I'm just going to, I was keeping up with the conversation, and there's a little bit of conversation that happened here, but if you skip down, there's a question from Kristen, if you want to pick up there.
Kristen, you mentioned that disease could not be assessed with eDNA, but pathogens have been assessed in some eDNA studies. Could Hake be used as a test model for assessing disease load? That sounds super interesting. I don't think of a reason why you couldn't do it. I guess the only concern, you'd have to make a primer for it, of course, for whatever disease you're interested in. You'd also be surveying the disease, not which species it was infecting. So if there's a Hake-specific disease, certainly that would be relatively straightforward. If you're worried about something more general, fish disease that could be in Hake or rockfish or herring, that might be harder to interpret directly. So I don't know a whole lot about disease. I do know that there are certainly disease primers and studies out there that are interesting, but we haven't thought about it. So good question. And there's a question for Ian. Acoustic index using the Pacific Hake stock assessment is for Hake's ages 2+, only with selectivity at age estimated from the age samples collected during the survey. Yeah, eDNA index seems awesome, but for assessment purposes is hampered by no way to estimate selectivity. Right, so this is a great question, Ian, and this is a thing that we've talked about. So there is an age 1 index for the Hake that's sort of in development from the acoustics team, and we've talked about trying to sort of do that related to the Hake survey or the eDNA stuff. I've thought about breaking the age structure up into a whole series of numbers, so age 2, age 3, age 4, age 5, etc., and figuring out how to lump them together or split them to talk about what their contribution to the eDNA is. It turns out that's hard and it's slow, and so love to chat more about how to go about doing that. But it's super interesting. Tracy 3, but you get lots more info than just for one species, right? I'm not sure I understand. Per sample. Yes, that's true. So yes, per sample. So yeah, we've run 3 species right now with the qPCR. We have some work with metabarcoding using these same samples. We're planning on doing some krill work. There's talk about doing, with these samples, there's talk about doing marine mammals with some of these samples. So yeah, so there's a sample collection side and then there's the application. Eventually we'll run out of sample, but we seem to have a fair amount left right now. And so there's lots of options for doing particular investigations. Okay. Oh, Anna asked a sample per laborer. Thank you, Anna. And Zach is answering a question. And Zach has a question. Last sample here. Last question I see. Do you think qPCR metabarcoding results can be integrated to address the compositional nature of metabarcoding to provide abundance estimates at scale? Yeah, I do. That's some hard math as well. Yes, I do definitely think we can do it. I sort of have a couple papers in the back of my brain called Making Barcoding More Quantitative, and I think I know how to do it. And I think there's a lot of power from moving away from qPCR into metabarcoding and sort of getting many species at once for total abundance. So anyway, very exciting. I think there's a lot of power here and a lot of interest, and would be happy to chat with anybody else who wants to ping me later.
Thanks, Ole. That was a lot of questions, and there was a lot of really great conversation here in the chat, which is good. We had another question that was coming in. Because we only have a couple minutes left, I encourage you all to please email Ole directly if you have additional questions, and I'm also going to drop in a new NOAA Omics email address if folks would like to follow up with me as well about the seminar series or anything omics related happening across NOAA. Okay. So with that, I think what we're going to do is start to wrap up the webinar here. So I just want to thank you all again for joining us today, and thank you so much, Ole, for the fantastic talk. It was super interesting, and I really like seeing combining eDNA with these more quantitative measures to quantitatively measure Hake abundance. I thought it was super cool. So for the rest of the group, the NOAA Omics Seminar Series takes place on the 3rd Wednesday of every month at 12 p.m. Eastern Time. So our next seminar is going to be on October 20th, 2021, and it will include a presentation from Sarah Shaw, who is a PhD candidate at Northeastern University. Her presentation is titled Evaluation of the Role of Inversion Polymorphisms in the Evolution of Sympatric Intraspecific Diversity, a Theoretical and Empirical Study. So I hope that you can all join us for that next presentation. And with that, we will end the webinar. Thank you, everyone.
Thanks for coming, everybody.